I'm really happy to introduce Dr. Daniel Promislow today. He got his undergraduate degree at the University of Chicago and his PhD in zoology from Oxford University. He then did postdoctoral positions with the French National Center for Scientific Research, and also with the Natural Sciences and Engineering Research Council of Canada and the University of Minnesota. He was a member of the genetics faculty at the University of Georgia for a number of years before joining the faculty at University of Washington School of Medicine in Laboratory Medicine and Pathology and Biology. He's published more than 200 peer-reviewed articles in major scientific journals and is a recipient of the Breakthroughs in Gerontology Award, or the BIG Award, from the American Federation on Aging Research. And his research has been funded by NSF, NIH, the National Institutes of Aging, and several private foundations. And I've been following his work for a number of years because I do have an interest in aging, but I mostly study birds, and that's just about the only critter you haven't studied aging in. So he's going to tell us about the dog aging project today. So welcome.
Thank you so much, and thank you for the kind introduction and the opportunity to share a little bit about the dog aging project with you. So just an outline of what I'm going to talk about. I'm going to start with a very brief overview of aging and aging research, why I think it's actually a really cool thing to study. And I'll talk a little bit about why, in some really important ways, the lab is different from the real world. I'm going to show you one mathematical equation. You can see it here, and there won't be a quiz afterwards, so you can relax. And then I'm going to talk to you a little bit about dogs and why they're cool, just as cool as aging research is. And then I'll tell you about the Dog Aging Project, what we hope to learn, and where we are at in our launch towards the complete running of the project. It's a huge nationwide project, and it takes a long time to set everything up. So let me start just with a very high-level view of aging. So I started out studying aging as a graduate student in Oxford, and I did comparative biology. So I was comparing patterns of aging in different species. I was just looking at mammals, and you can see all of these mammals on the top. Lots of different kinds of mammals, from relatively short to extremely long-lived. And bats, for their size, are probably the most long-lived mammal. They're not the longest lived, but for their size, they probably are. And then across the whole world of animal diversity, you get really short-lived animals like mayflies. The adult stage is a day, 24 hours pretty much, all the way up to this clam here, which as far as we know is the longest lived animal species. Individuals have been found in the ocean that were 500 years old. And then of course, we have trees that can live into the thousands of years. So one of the reasons aging is so cool to study is that it captures biological diversity as much as any other trait that we might measure. The other thing that is really kind of mind-boggling about it is that you can study aging across huge physical scales, orders and orders of magnitude. So we can think about what happens in terms of aging, measuring small molecules. So you can measure you think about the small molecules like ATP in the Krebs cycle, those molecules change in quantity as we age, and the flow through those paths change as we age. And we can study things all the way up to blue whales and California redwoods and how those things age. So no matter what spatial kind of framework you get excited about, whether it's the really nanometer microscopic all the way up to the ecological, there's something for you in terms of aging. And of course, in our own lives, we can think about how aging impacts us. So we know that most of the major killers in human populations are age-related diseases. You think about heart disease, cancer, neurodegenerative diseases like Alzheimer's disease or Parkinson's disease. These are all diseases of aging. Of course, if you ask people what are the risk factors for these kinds of diseases, they can usually guess pretty well, even if they're not biologists or people who study aging. We know, for example, that smoking, and alcohol intake, a poor diet are all associated with heart disease risk, with cancer risk, even with Alzheimer's risk. There's one gene that we know about called ApoE. It has a few different forms, and there's one particular form that we call the epsilon 4 allele or form that's associated with a higher risk of Alzheimer's disease. But here's the thing. If you ask, well, what about age as a risk factor? So many of you who are listening to this are in your late teens or early 20s, and your chances are you are at extremely low risk for any of these, heart disease or cancer or Alzheimer's disease. So if you compare these to aging, and I'm going to show you plots now where we look at the relative effect of aging compared to these other risk factors. Those other risk factors are still here. You can sort of see them sneaking up in heart disease. You can barely see them in cancer, and you don't see them at all for Alzheimer's disease. Aging is the #1 risk factor for all these diseases. So if we can understand something about how it is that aging leads to the increase in risk for these things and ways to slow it down, the hope is that we can delay the onset or decrease the severity of the effect of these diseases of aging. And so not necessarily make people live longer, but help people to live healthier longer. So some people would argue that we've solved the biology of aging. We know that there are all these hallmarks. This figure is a now very highly cited figure from a 2013 paper from Lopez-Otin and colleagues that came out in Cell. And it includes a long list of things that we now know happen as organisms age. And almost every organism, yeast, which of course are fungi, and nematode worms and flies and mammals, all of them show these various hallmarks of aging. Mutation or genomic instability, telomeres get shorter as we get older. There are epigenetic changes. That is, the structure of the chromosomes, the chromatin actually changes as we age. We lose the ability for proteins to form the appropriate structures. I won't go through the whole list, but we know a lot about what happens as we age. And many have argued that if we can slow or reverse these processes, maybe we could slow or reverse aging. And in that regard, people have tried to do that, both genetically. So we've discovered in yeast and in nematode worms, and fruit flies, and in lab mice, that if we alter these genes, we knock them out, or in some cases increase expression, we can make these organisms live longer. Also, there are many drugs that people who study the biology of aging have discovered, and lifestyle changes that make these species live longer. Probably the one we've known about the longest is dietary restriction. So if you feed a rodent or a fly or a worm about 60% or 70% of what it would normally eat if it were allowed to eat as much as it wants, it lives longer and healthier. And there are many drugs. Probably the most successful one is rapamycin. This is a drug that's given to solid organ and bone marrow transplant patients to help prevent rejection. At much lower doses in these lab species, it makes them live longer. And I'll come back to that when I talk about the dog aging project. So we know a lot about what happens as organisms age. We know about the genetic pathways that seem to be involved in the lab. We know about interventions that seem to be able to make organisms live longer and in some cases healthier in the lab. But here's the big challenge. So the lab is not the real world, and people are more variable than the worms and flies and mice that we study in the lab in two important ways. And this is illustrated by this now very old photograph from 1914 of cadets lined up at what was then Central Connecticut State College. So these are military cadets lined up from shortest to tallest in rows, each of which is an incremental inch, from about 5 feet to well over 6 feet. And you can see they form a pretty nice bell-shaped curve or a normal or Gaussian distribution. And that's true of just about anything that we measure in the real world, whether it's in people or in other organisms, that there's a lot of variation, and often it's this nice normal distribution. And I warned you I was going to show you one equation. Here it is. So all of the variation that you see in this picture is due to two and only two factors. There's genetic variation, there's environmental variation, and there's the interaction between the two. So the effect of a gene might depend on the environment. So for example, if you have a gene in plants that alters plant height, that a particular allele might make plants taller in the shade, but actually make them shorter in the sun. So the effect of a gene can depend on its environment. And so what this is saying is that the variation in P or phenotype is explained by the variation in all our genes, our environment, and the interaction between the two. When we study organisms in the lab, we try and make environmental variation 0, and we try, if there's any genetic variation, to make it so that there's just a single gene that we manipulated, everything else is fixed. So it's not very much like the real world. So the question is, how can we understand variation in the real world? That's the challenge. And that's what we work on in my lab. Most of what we do physically, actually, in the lab is on fruit flies. I'm not going to tell you about that today, just going to tell you about dogs. But we really are trying to do the same thing with fruit flies in the lab and people's pet dogs. So here's the challenge. We're trying to understand how genes in environment shape phenotype or traits. In our case, aging and age-related disease. So we have lots of genes that could potentially affect the downstream traits, and they might be interacting with each other in a complex network that we have to tease apart. But I want you to notice that the distance between the genotype and the trait is huge. So just understanding the genes is not going to explain the variation in the traits that we're interested in, like how long an organism lives. In between the genes, the genotype and the phenotype is what we call the endophenotypes, or the omics, or the systems biology. So the epigenome, what the chromatin looks like, the transcriptome, how much each gene is turned on or off, the proteome, how much of each protein is produced, the metabolome, which is all the small molecules that are circulating in the blood, and the microbiome, all the microbes. We think of the microbes mostly in our gut, but in fact, every body part on the outside of our body has its own unique set of microbes. And these are actually really important. They help us stay healthy. Without a microbiome, we couldn't digest our food properly. In my lab, we focus mostly on the metabolome, which is sort of the last step between genome and phenotype. The metabolome makes up all the building blocks. The functional building blocks, I mentioned the Krebs cycle or the TCA cycle. Think about all those molecules that you learn about, in biochemistry that you had to memorize, that determine how glucose is turned into energy, for example, or how proteins are made. All those small building blocks, the structural building blocks as well, like the amino acids that make up the proteins, are part of the metabolome. So that's what we study, really to try and bridge the gap between the genotype and the phenotype. So that's my perspective. I'm interested in variation and really completing the genotype phenotype map to understand why we all look different. And of course, many of these features like the metabolome, the arrows here are pointing downwards, but metabolites like methyl groups can bind to the genome and affect the transcriptome. So there's lots of flow in this network. So the equation, we can turn the equation into a picture that explains the dog aging project. So the dog aging project that I'm going to tell you about today is really an effort to understand how genetic variation in dogs and environmental variation in dogs can explain variation in aging in dogs. And we can add a little bit more information. So some of the environmental variation is responsible for extrinsic mortality in dogs, whether that dog lives on a busy street or not as part of its environment. And from an evolutionary perspective, how the genome changes over hundreds and thousands of generations depends on how genes influence survival, which is measured by aging. So that kind of completes the loop. Okay, so I've told you already that, so the goals of the Dog Aging Project are to identify the genetic and environmental determinants of healthy aging in companion dogs, and also to determine whether we can increase healthy aging or healthy lifespan in companion dogs. So when I say companion dogs, I mean the same thing as pet dogs, dogs in people's homes, not dogs in labs. The pictures that you're looking at are pictures of Frisbee. Frisbee is sleeping next to me on the floor, which is mostly, right now as I give this talk, which is mostly what she does now. She's 15 and a half years old. She's about 45 pounds. She's very old. She is a geriatric dog. In her younger days, this is Frisbee hiking up a mountain outside of Seattle. This is Mount Rainier in the distance. And this is Frisbee looking back because she's running ahead. She was always running ahead and then waiting for us slowpokes to catch up. This, in fact, was just 4 or 5 years ago. And part of why dogs are a powerful model to study aging is because they age so quickly. Frisbee now is 15 and a half. She struggles with pretty serious osteoarthritis, just like people get. She struggles with very poor pancreas function. So I actually now am one of those dog people who make their dog her own food. Once a month, I make a month's worth of dog food and put it in the freezer so that she can digest properly. The Dog Aging Project actually started back in 2007 when I was at the University of Georgia. And it was there that I met Kate Creevy. Here we are a few years later, and here I am a few years younger, not quite as gray. And then I moved to the University of Washington in 2013 and met Matt Kaberlein. Here he is with 2 of his dogs, and me again with Frisbee. And the 3 of us really formed the leadership group of the Dog Aging Project. One of the important things about the Dog Aging Project is that it's really team science. There's something really satisfying about working alone in the lab and making a discovery all on your own. There's something even more exciting about recognizing that you can't possibly know everything there is to know. But if you surround yourself with people who in the aggregate do know everything you need to know, it's a pretty exciting way to do science. So it's very much a collaborative project. And if there's sort of a first take-home message, it would be the power of working together. So why do this in dogs? So I already alluded to this, that the lab is a really awesome place to make some amazing discoveries. And so much of what we know, including things that we know about humans, we learned from these lab organisms. People often say, well, why bother studying the fly? A mouse, we can see it's a lot like us. It's got vertebrae and a blood system like ours and a brain that is a little bit like ours. But a fly? Well, in fact, there's a lot that we know about human health and disease from flies. For example, just about everything we know about the earliest stages in human development, when genes turn on that tell the developing fetus where to put its arms and where to put its legs and where to put its fingers, that whole signaling cascade we learned about from fruit flies. So these organisms can teach us a lot, but there are things that they don't teach us. First of all, as I told you already, they usually exclude variation. Secondly, in mice a little bit, but certainly not in worms and flies, there isn't a lot of pathophysiology. We don't know why a fly dies. We don't have a healthcare system to carry out diagnosis of a fly disease and then give it prognosis, and certainly not treatment. And no one ever comes up to me after I give a talk about fruit flies and says, oh my goodness, I love fruit flies. So that's not, so the dog really addresses all of these things. There's variation. There's lots of pathophysiology. Dogs have as sophisticated a healthcare system almost as we do. And people really do love dogs. So how a research career unfolds is really an interesting thing, and something I would be very interested. I know many of you on this call are early in your own careers and wondering what it is that your path is that lies ahead, and how do you figure out what the right path is for you, and how do you get there? Sometimes you don't know until you get there. So I spent my graduate career studying mammals, and that was back in the late 80s. And then starting in the mid 90s, I started working on fly aging. And in 2007, this paper came out. And I didn't know this paper was going to come out, and I didn't know that everything that I had been studying was kind of setting the stage for me to read this paper in a different way than I might have otherwise. This paper kind of rocked my world for a couple of reasons. First of all, I love dogs. Dogs are awesome. Look at that variation. So this is a Chihuahua and a Great Dane walking side by side. Between the smallest and large breed dog, size varies by about 50-fold. In humans, it varies by a factor of 2 or 3. So the amount of variation in dogs is huge, and lifespan is huge. And this is something that I already knew, that those big old Great Danes are super sweet, but they don't live very long, whereas that Chihuahua can stick around for a long time. The other thing that was really striking to me was this gene right here, IGF-1. It's the insulin growth factor 1 allele. Turns out, usually when we try and map traits, and I sort of alluded to this when I showed you that picture with the layers, and I said the genotype is far away from the phenotype. I'm not going to talk about it today, but when people try to map traits in humans, for example, body height, they find dozens and dozens of genes that are associated with body height, but each one of them explains a tiny, tiny amount. So if you have 2 different alleles of a gene, depending on whether you have the tall allele or the short allele, might impact your height by a fraction of a millimeter. And here we're talking about a gene that has effects on the order of maybe 10 or 20 kilos, huge effect. And the other thing that's notable about this gene is, because I work on aging, I knew that many studies in worms and flies and mice and even in humans suggest that the insulin growth factor, insulin signaling pathway appears to be associated with aging. And these large breed dogs have a certain type of this gene, and they're short-lived. So that got me interested, and I'm wondering whether there might be something special here. And I decided to start working on aging in dogs. So one of the really striking things about aging in dogs is this, and here you can see it illustrated. So in mammals in general, the larger species, here we see size on the x-axis on a log scale. So going up, so these are in grams, from tens to hundreds of grams all the way up to millions of grams. And the larger species are longer lived. And the same is true in birds as well, which are also on this figure. The birds are in blue and the mammals are in red. And so you can see that larger species tend to be longer lived. Here are the bats, which are that really cool outlier, very small but long lived. But look at the dogs. There's a lot of variation, but in general, you can see that the large breed dogs are shorter lived than the small breed dogs. So I knew that, and that got me thinking, maybe we can work on dogs and figure out why it is that large dogs are short-lived. So the other thing is that dogs are incredibly variable. So these are just a few of the breeds out there. from small dachshunds to huge Irish wolfhounds and funny looking Shar-Peis that have the wrinkly skin. Lots and lots of variation, all one species, all evolved from the wolf about 15 or 20,000 years ago. So if you go to the dog park or just go for a walk and you see other dogs, you can see all that variation. But there's also variation in disease risk. And this is actually the first paper that we wrote was exploring that variation. And what I'm showing you here are the percent of deaths due to one of 4 different causes in 4 breeds. So first of all, you see in blue, respiratory disease. This is the bulldog. This was the mascot at the University of Georgia, where I spent 18 years. And bulldogs, like many brachycephalic or flat-faced breeds, struggle with respiratory diseases, and they often die of those. This Chihuahua is most likely to die of heart disease. Retrievers, this particular breed of retriever gets a lot of leukemia and lymphoma, and retrievers in general get a lot of cancer. Golden retrievers, about half of all deaths are due to cancer. And then again, the Shar-Pei with the wrinkly coat, they struggle with stomach and intestinal disorders as well as skin disorders. So huge variation. So that's that genetic variation that I talked about, and the phenotypic variation. Of course, they also have environmental diversity. So there are about 90 million dogs in this country, and they live in wealthy environments and not so wealthy environments. So in the top left, you can see a dog in front of its doghouse. On the bottom left are dogs on the street. There are healthy dogs in the top right, that dog going for a hike in the mountains, and on the bottom right, dog smoking along with its owner. And of course, importantly, there's also owner diversity. So people of all ethnic backgrounds own dogs, people of all socioeconomic levels own dogs. So that environmental diversity is important too, to help us understand how the environment shapes aging. So taken together, there are really 3 things that motivate the dog aging project. There's the power of diversity, and a colleague in my lab said that dogs are to animals what broccoli is to plants, and who doesn't like broccoli, right? So these are all the same species: broccoli, purple cabbage, cauliflower, kohlrabi, kale in the background, Brussels sprouts, all one species. So dogs are kind of like that, incredibly diverse. And for us, there's tremendous power in that diversity. It gives us something to explain. There's also the power of paying attention. So 95% of what we're going to do in the Dog Aging Project is just really carefully pay attention. And of course, we're not the first to do this in this country. Probably the first large-scale, long-term study that was really a study of paying attention is the Framingham Heart Study. It started in Framingham, Massachusetts in 1948. The people who are still enrolled in that study are now the grandchildren of the people who were adults in 1948. And we've learned a lot from this study. For example, we understand the relationship between smoking and risk of heart disease because of the Framingham Heart Study. The first study that was explicitly about aging in general was the Baltimore Longitudinal Study of Aging in Baltimore, Maryland. The prostate-specific antigen study came out of this long-term longitudinal study. Again, just the power of paying attention. And we're not the first people to do this in dogs. So in 2013, the Morris Animal Foundation started a study of 3,000 golden retrievers. So this one is just about golden retrievers and primarily about cancer. Ours is about all dogs and all causes of aging. But what we learn is mostly just from very carefully watching what happens to individuals over time in what we call a long-term prospective study. And finally, the power of love, that people love their dogs. Every single individual in this picture can tell you a lifetime of stories about their relationship with their pup. And that really motivates what we do. People are so keen to sign up for our project and share their stories about the health and life experiences of their dog. So that gets me to the dog aging project. And so I'm going to tell you a little bit about what we're doing, how we're doing it, and we've really just begun. So I'll tell you about some of our very first discoveries, but this is really just the beginning. And I welcome all of you to follow us. If you have a dog, sign up. But if you don't, just follow us and even join us in analyzing data over the years. So the way things start is you go to our website, which is just dogagingproject.org, and you click on the Nominate Your Dog button. Tell us what your email is, what your zip code is, how old your dog is. And that's the one thing, you have to know how old your dog is, plus or minus 6 months, because if you don't know that, it's hard for us to study aging in the dog. So as soon as you nominate the dog, you'll then get an email inviting you to create your personal portal where you will share lots of information about your dog. And the first information that you'll share, which will take 2 to 3 hours, is what we call the HLES, or Health and Life Experience Survey. So it's a detailed survey that tells us all about not only how old your dog is, but its health status, what kind of diseases it's been diagnosed with over its entire life, its diet, its age, its sex, whether it likes to chase birds and squirrels, really everything that you could imagine asking about a dog, we ask. Our goal is eventually to have 100,000 dogs in the pack. We're well on our way there. Last week, we passed the 29,000th dog, so we're pretty excited about that. And these dogs are part of a longitudinal study. That means that this is the first year, but we're going to ask them information every year for the life of the dog, and that makes it longitudinal. This includes, our first set of dogs include young dogs and old dogs, males, females, all breeds, mixed breed dogs, everywhere in the US. Within this set of dogs, we then choose a subset of dogs to send sample kits to. We'd love to do it for all, but it's just way too expensive. So the first is what we call our foundation set of dogs. And these dogs will have the survey data. They will also have uploaded electronic medical records from their veterinarian, just like human electronic medical records. We'll send them a cheek swab. They swab their dog's cheek, put it back in the mail, pre-addressed kit, and it goes to our sequencing facility for whole genome sequencing. The second group we call the precision group. They get everything Foundation gets, plus once a year they take a kit that we send them to their veterinarian for collection of things like blood, hair, fecal samples, urine samples. All of those then get sent to our clinical lab for processing, and then on to other labs like my lab for molecular biology. So in my lab, for example, we'll analyze the metabolome in the plasma. Another lab will do the fecal microbiome through sequencing all the bacteria and so on. A fourth group, the intervention group, will be a small subset of all of these dogs enrolled in a clinical trial of rapamycin. I'm going to tell you a little bit more about that. And these dogs only are taken by their owners to veterinary teaching hospitals. And then finally, there's a separate cohort of about 300 dogs that are super old dogs, 1 in 1,000 for their size. So the oldest 10-pound dog is going to be a lot longer lived than the oldest 150 or 200-pound dog, obviously. So here's that figure again, and I'm just going to fill in the blanks a little bit and just to tell you a little bit more about how all this fits together. So the genetics is breed information that the people share with us, and then for 10,000 dogs, and even more if we can raise more funds, whole genome sequencing. For the environment, it includes things like air quality, water quality, green space. So the Environmental Protection Agency actually has tons of publicly available data. So if you were to tell me your zip code, I could tell you in your zip code, compared to every other zip code in the country, levels of nitric oxide, sulfur dioxide, carbon monoxide, particulate matter from diesel exhaust, all of this information is known. So we match that to each dog along with information about the home environment, diet, exercise, healthcare, and so on. Downstream of that are all the measures about aging in the dog. We'll even teach people how to do cognitive tests in the dog. And then finally, in that middle group, the bridge between the genetics and environment and the phenotype are all the molecular biology. And so this really creates a complete picture of aging in dogs. And the Dog Aging Project is actually 4 projects. So the first project is just studying aging in dogs. So the amazing thing about veterinary medicine is that, like human medicine, there are all these specialties. So as in human medicine, you could, if you needed to, for your dog or cat or other pet animal, you could go see a veterinary ophthalmologist for an eye problem, or a veterinary orthopedic surgeon for a broken bone, or a veterinary oncologist for cancer. If you were to take a very old friend or relative to the doctor, you might go see a geriatrician who specializes in human medicine for older people. There is no geriatrics in veterinary medicine. So really the first project is creating the information about what's normal aging for a small breed dog or a large breed dog, to create that kind of veterinary geriatrics. The second project is to link genetic variation to the variation in aging. The third project is what we call the systems biology, to link genes and environment to phenotype, so we can not only detect what genes and what environmental factors make dogs live healthier longer, but what are the mechanisms by which they do so? So if there's a particular diet that helps dogs live longer, how does it work? This will help us figure that out. And then finally, project 4, we call the TRIAD trial, which is an acronym for Test of Rapamycin in Aging Dogs. So I'll tell you a little bit more about the detail of each of those and some of what we already know. So one of the striking things about dogs is how similar they are to us in many ways. So what we're looking at here are some data that we published just a few years ago. This is work by a former graduate student from my lab, Jessica Huffman. And what she showed is that the proportion of individuals dying of a particular cause of death at any age has a particular pattern. So for cancer, people in their early years have a pretty low risk of cancer. By the time we reach 50, our probability of mortality is still pretty low, but of those people who die, about 40% die of cancer. And then as we get older, the proportion of people dying of cancer at a particular age falls as the proportion dying of heart disease and neurodegenerative diseases goes up. So similar pattern in dogs. There's an increase in the proportion that die of cancer, and then it declines. And similarly for death due to poisoning and drug overdoses, general trend is a decline with age in humans and a decline with age in dogs. There are some important differences though, and the biggest one of those is probably cardiovascular disease. Dogs get heart disease. They get mitral valve problems, they get cardiomyopathy, so heart function problems. They don't get vascular disease, clogging of the arteries, for example. So the proportion of deaths due to cardiovascular disease, so this is heart attack and stroke, steadily increases in humans. Dogs tend not to get these things, and what little they get doesn't increase with age. So some really interesting differences, which potentially gives us an opportunity to study why it is that dogs are protected from these deaths. There's also this striking variation in dogs, which we don't see so much in humans, and that has to do with inbreeding. In general, people are quite outbred. In dogs, of course, the purebred dogs, which here are shown in blue dots, are much more inbred than the mixed breed dogs. So what we're looking at here on the left is life expectancy at the age of 2. So if you're 2 years old, how many more years can you expect to live as a function of weight? And you can see this pattern, the steady decline with age, larger with size, larger dogs are shorter lived. But also this big difference that mixed breed dogs for a given size tend to live longer than purebred dogs. So Frisbee is right here in this size class. She's about 18 kilograms. She's a mixed breed dog. She's a mix of Chow, German Shepherd, and Cocker Spaniel with a little bit of Golden Retriever and some other stuff thrown in. Because I'm in the business, I've actually done 3 different tests with 3 different organizations, ourselves, as well as 2 companies, and it all came back the same. So her total life expectancy, given that she lived to 2, was about another 10 years, and she's up here, way up here. She's 15 and a half, so pretty long-lived. The other thing you notice, so there's variation between breeds. Some breeds are actually longer lived, partly because they're more outbred, and certainly the mixed breed dogs. But even within breeds, it's a weak effect. But what this is showing you is the distribution of age of death for outbred golden retrievers in blue and inbred golden retrievers in pink. And there's a little bit of a benefit for being outbred. So mixed breed dogs live longer, small dogs live longer, and in the golden retrievers, lifespan is shorter for those that have a higher coefficient of inbreeding. So just going to quickly run through the projects in a little bit more detail. I'll tell you a couple, and then I'll tell you a couple of results that are sort of the first results rolling off of the study. So as I mentioned, Project 1 is really creating geriatrics in dogs. And these 3 concepts are 3 really important human concepts. And that's why I wanted to talk just a little bit about them. So in understanding what happens as we age, if you think of your older friends and family members, they struggle with, so they're likely as they get older to struggle with a couple of different things. One is what we call multimorbidity, the coexistence of 2 or more chronic diseases. So as we get older, the likelihood of having 2 or more chronic diseases goes up. And that creates some real challenges as we're trying to address the problems of one disease that might exacerbate the problems of some other disease. And the more chronic conditions that we have, the higher our risk of death. And that can really have a negative effect on quality of life. There are also struggles with frailty, that even if we're alive, if we struggle with frailty, functional, cognitive, physiological decline, that can get in the way of quality of life. And all of these add up to what we call health span. So health span is the period of life of normal activity, social engagement, free from disability. So what we're really interested in understanding is not how to get to a long lifespan, but how to have a long health span, how to maximize this period of healthy life. So projects 2 and 3, as I mentioned, are really trying to understand the genes that influence that health span, and then the systems biology, the mechanisms that link genes to aging. And it's just one example of what the systems biology might look like. So what we're looking at here is what we call the epigenome. So the genome you can think of as the stretch of, in us, 3 billion base pairs, which make up about 15,000 genes that code for all the proteins and the RNAs that make us who we are. Partly, anyway, make us who we are. The other thing that's important in the genome is not just the sequence of A's and C's and T's and G's, but the shape of the genome, whether it's really compacted chromatin, or what we would call heterochromatin, or more open euchromatin. The more open it is, the more likely genes are to express. And that structure of openness and closeness is unique for each cell type. That's partly what determines whether a cell is a liver cell or a brain cell or an immune cell, is the epigenome. We can measure that in a couple of different ways. I'm not going to go into the details other than to say that we took about 80 dogs in a pilot study. We took their peripheral blood mononuclear cells, or their white cells, B cells and T cells, and we measured the methylome, and we measured ataxic, and what each dot shows you is a dog. And we created a model that allowed us, from this information, to predict how old that dog is. And you can see that we did a really good job, that the predicted age is close to the observed age. Now the big question, and this sample size is really too small to ask, but this is what we want to do in the Dog Aging Project. We want to ask, if you look older in terms of your epigenome than you actually are. So this dog here is 7 years old, but it looks like it's 9 in terms of this epigenome profile. Does that mean that it's a sicker dog or a shorter lived dog? We don't know yet, but in humans, people have been doing this, and it looks like often that is exactly the case, that the epigenome is a prediction not only of your chronological age, so the age that I would calculate if I looked at your driver's license, but it's also a measure of your biological age, sort of your inside age, which is associated with how healthy you are now or in the future. And I'm going to skip this and move right along. And then I just want to tell you about Project 4 briefly. That's asking can we actually intervene in a way that increases the duration of healthy lifespan in dogs? And that's where rapamycin comes in. Rapamycin was a molecule that was discovered from bacteria that were dug up in the soil of Easter Island, which is where this statue is. Easter Island is also called Rapa Nui, which is why we call it rapamycin. This was discovered back in the 70s. It's an FDA-approved drug that's used in transplant patients, and it's the most effective pharmacological intervention in laboratory animals in terms of improving lifespan, brain, heart, muscle, immunity, and so on. So TRIAD, so the first and foremost thing is safety. We don't want to do any harm to dogs, and to make sure to help us that our decisions are the best possible, we have an animal welfare advisory board. And then if you do a clinical trial in humans, the National Institute of Health will set up an independent data and safety monitoring board. So you're not allowed to see your data until the study is finished, but the monitoring board sees the data, and if there's anything going wrong, they'll tell you if you need to shut the study down. If it's so good that it would be unethical not to give the drug to everybody, they would shut it down and tell you that. So this study is going to be randomized. So dogs are either given a placebo or rapamycin. We won't know which. All genetic backgrounds, pure and mixed. Middle-aged, large breed dogs. Okay, so just the last 5 minutes or so, I'm going to tell you where we're at. So this is the launch, which initially we launched last spring, and last spring was also, obviously, as you know, when we suddenly found ourselves in a bit of a lockdown. And so we had to change some of our targets. And fortunately, we figured that out. And the next catch was quite successful. And that's what I'm going to tell you about, what we've been doing in the past year. So we haven't yet been able to get many dogs coming into the clinic. We're just finally starting that now. But the launch has been a tremendous success and pretty exciting. So the official launch actually was in November of 2019, where we first got people to nominate their dogs. We did a big media launch, and we had a lot of hits around the world on various sites, radio, television, newspapers, magazines, and the internet. Almost overnight, we had 50,000 people nominate their dogs, and that number is now over 90,000 people that have nominated their dogs. They've all been invited back to complete the health and life experience survey. And so during the past year, we've all been on Zoom meetings, our team of about 70 people, building the infrastructure to create a dog aging project. And it's not only a community science study in the sense that the community of dog owners like you are the scientists out there sharing your science information about your dog with us. It's also an open science study. So everything that we collect, all the information we collect, we will share with the community, and building a platform that allows us to do that and will allow us to continue collecting data for decades to come, not just with this generation of dogs, but many generations in the future, has been our focus this year. We have used the time also to communicate with our participants, not only about dogs, but also about things relevant to COVID. People asked us lots of questions. Can dogs get COVID? And what about veterinarians in a time of COVID? Initially, all the veterinary clinics closed, and then they reopened and are busier than they've ever been because of all the dogs that were adopted during the pandemic. So people are able to access a summary of the data. If you'd like to go and look yourself, you can just go to data.dogagingproject.org. And this just is a little bit of a slice of the data. This is our official release from December 31st of 2020. We're now at more than 29,000. This was 27,500. And this just shows you there are purebred and mixed breed dogs, about half male and half female. And we ask them all kinds of things. So for example, do your dogs chase squirrels and rabbits from never to always? Or what's your heating fuel source? This could be important. If you live in a home that's heated by wood, how does the particulate matter from the burning wood impact your dog at all? Nobody knows, but we will find out. Importantly, what kind of food does your dog eat? Is it kibble, which is the majority, or what about home-cooked food, raw food, and so on? How often does your dog eat? And then, of course, we ask a lot about health. So of the 27,500 dogs in this particular group, 22,000 said that something had gone wrong with their dogs. The most common is skin disorders, all the way down to rarer things like blood cancer, for example. And this just is a little bit detailed. So for those almost 8,000 people who said trauma, we then ask, well, what kind of trauma? And the most common is a bite wound from another dog. And then we would ask, how was it treated? How long ago did it happen? What was the outcome? Were there any other adverse events, and so on? And so just to finish up, let me tell you a little bit about where we're at. So we've now got this huge amount of survey data, the most comprehensive survey data that any public science study has ever had on dogs. And so we're starting to ask questions. And so, of course, an obvious question, so neither of these are Frisbee, but I spent a lot of time doing this with Frisbee. So I thought, what happens to walking speed as dogs age? So I plotted these data, and sure enough, as dogs get older, they slow down from brisk early in life to slow later in life. Now, a couple of graduate students on our team have been digging a little deeper, and by collecting all the information that we do, we're able to look in more detail, and some really cool things are beginning to emerge. And this is just a tiny little sliver of all the questions that the team is now working on. There are about 15 different papers that people are writing as I speak, and 12 months from now will I hope, all be published. So let me just go in a little bit deeper here. So what these two graphs are showing is not dog speed, but activity duration. How long each day is the dog active? And so this is in hours. It's plotted on a square root. So you see the little square root. So you can see the distance between 2 and 3 is bigger than the distance between 3 and 4. Don't worry about that. What's kind of cool is that as dogs get older, they spend less time outside. That's certainly true of Frisbee. I walk her, mostly she wants to lie down and sleep. I walk her twice a day, 20 minutes each walk, and 20 minutes is enough time for Frisbee to get around a full city block here in Seattle. And mostly while we're on the walk, she just wants to smell everything. She doesn't see so well. I'm not sure she hears at all, but she sure does smell well, and that gives her a lot of joy. But there's some other cool things here. So older dogs spend less time outside, active. But if you look at the colors, and I apologize if anyone here is colorblind, what this is showing is owner age, and this is kind of cool. So this is the line for 25 to 34 and 35 to 44, brown and dark green. And then up here in the magenta and the purple is 65 to 74, 75 and older. In general, older people actually spend more time outside with their dogs than younger people. Initially, we thought it would be the opposite. And then we thought, well, these young folks are starting their careers and they are busy, and these older folks, many have retired. Interestingly, this red line is sort of the outlier. These are people between 18 and 24, and they're likely to have more free time than people in their 30s and 40s. So that added a dimension that we'd never thought about. And then the other thing that in hindsight makes sense is that rural dogs in blue spend a lot more time active than the green and red dogs, the urban and suburban dogs. And then finally, this is just to show that cancer reported by our participants goes up with age and is more common in large breed dogs in purple than in small breed dogs in red, which we already knew, so that's good. Reaffirms the quality of our data. So what this is showing is the probability of a dog having ever had infectious disease. And so it's a flat line because dogs that are 15 never get infectious disease, but they did about 15% of dogs get infectious disease when they're young and then not many thereafter. So that's why it's a flat line. Okay, so where do we go from here? Well, I told you about the environmental data. So now all the survey data and all the environmental data are available on our platform, which is in the Google Cloud, available for people to access. We are just shipping out, we've shipped out over 600 of the 10,000 total kits that we'll be shipping out for whole genome sequencing. We're piloting the kit data for molecular data. We're starting to move into the longitudinal phase, people filling out the second survey. And we're aiming for a public data release sometime in 2021 where we'll take all these data and push them out to anybody who wants to look at them. And that's the last point that I really want to make. The outreach potential for this study is huge, not just with the scientific community, but we're really excited about educational outreach. There are many schools, from elementary schools through high schools, through community colleges, that don't have the resources to create biology labs. We can create modules that are appropriate for students at different levels, whether they're in 4th grade or in a community college, to create labs that allow them to work with our data and actually to do original research and write papers. We're also really excited about how interdisciplinary this study is. People can ask about, for example, the effect of socioeconomic status on health, which is a big question for humans, maybe also for dogs. Climate change, how does a warming climate alter the prevalence of vectors that are transmitting infectious disease? Questions around ethics. Dogs can't give consent, only the owners can. What are the ethical implications, and so on. So really the scope of this project is limitless. And finally, I just want to thank our primary funders, the National Institute of Health, as well as other organizations that have provided funding for the project, folks on our external advisory boards, cardiology teams at veterinary colleges, veterinary teaching hospitals. And finally, I want to thank you for your attention, and I look forward to chatting with you more about this.
Thank you.
So, Terry, you want to manage questions? I see one. Once you ask one, they usually start flowing in.
Yeah, so there's a question here, is like, is the project present on any social media platforms where followers can easily keep up to date on news?
Great question. So we have Twitter at Dog Aging Project, and we have a Facebook page, which is the Dog Aging Project Facebook page, so you can keep up there. You can also sign up for our newsletter. You don't actually have to be a member of the PAC to get the newsletter. We send out about 80,000 newsletters every month. So there are lots of ways to hear about the project. And then we have a frequently asked questions site on our website, dogagingproject.org. And for participants, we want to make sure that we make it as easy as possible for people to participate. So we have a team in College Station, Texas. That's where our communications team is based, and our Chief Scientific Officer, Kate Creevy. Dr. Creevy is at the Veterinary Teaching Hospital at Texas A&M. And so when people have questions, they email the Dog Aging Project and they get personal replies. Sometimes they'll even get a phone call if they're trying to understand something. So communication, your question is a really good one. And communication is so primary to what we do, not just to the participants, but to the general public.
Great. And Yeah, my dog Maggie was she was pack member 3,710.
Excellent.
Yeah, and so yeah, the communication as being part of the pack is I get, emails and things like that. And I also got this very nice card from y'all after Maggie passed, which was very comforting to get. And so here's another question. How do you pick which dogs get put into different test groups?
That's a really good question. So the first, I told you about 5 groups. So PAC, Foundation, Precision, Intervention or Triad, and Centenarian. So everybody gets to join the pack. All they have to do is know how old their dog is and complete the survey, which takes 2 to 3 hours. From there, we have a set of stratifiers. So to answer the scientific questions, we need as much diversity as possible. And so we have a set of different stratifiers. So there's size, age, sex, geographic region, and whether the dog is intact or not, whether it's sterilized or intact. That last one is important because dogs that are sterilized live a little bit longer and they die of different things than intact dogs. But really size, sex, age, geographic region. And so taking those stratifiers together, we create all these buckets. So we have dozens and dozens of buckets into which we want to put a certain number of dogs of each age and size and sex for a particular geographic region. And so the process by which we assign dogs is somewhat random in that we, having created that goal of a particular set of dogs in a particular bucket, the computer then starts choosing them and putting them in that bucket. One of the challenges that we're going to face is a psychological one. So in general, people are happy when the rewards that they receive exceed their expectations. So for those of you who are students, if you're studying for an exam and you expect, does Louisiana Tech have plus minus grading?
No.
Okay, so let's say you're studying for the exam and it's a really hard class and you've been really working hard, but this is just the hardest class you've ever taken. And if you get a C, you're going to be really relieved. That C is going to be pretty good. You get your exam back and you get a B. And you're just over the moon because your reward exceeded your expectation. On the other hand, if you're in a class and you think this is one of the easier classes that I take, and for whatever reason this stuff really comes easy, maybe you think it comes so easy you didn't study enough, but you really think you're going to get an A and you get a B, you're going to be really bummed. Same grade, very different feeling. So one of the challenges that we face, and this is sort of an insider perspective that I'm going to share with you, but I wouldn't put this on Twitter. One of the challenges is that people are excited to get their DNA kit and they're going to talk about it. And that's going to create an expectation. Right now in 29,000 people, well, a DNA kit for us costs about $100. Well, take 29,000 and multiply it by 100. That's a lot of money. We don't have that much money. So we can't choose everybody. And that's one of the challenges. And one of the reasons that even though we have a big grant from NIH, we continue to fundraise. One of the things we want to be able to do is eventually allow anybody who wants to go into that group. So that's sort of a long way around the answer. And the last thing I'll say about the TRIAD study, the dogs have to be large breed and middle-aged. And the primary reason there is that if we let 2-year-old Chihuahuas enroll in this study, we'll never be done because they're going to all live for another 15 years. We can't wait that long. So large breed, middle-aged dogs, we expect about a third of the dogs will die in a 3 to 5 year period. And also those dogs have to be near veterinary teaching hospitals and they all have to consent. And there's a very extensive consent process to make sure that these people really want to be in this one intervention study that's in the Dog Aging Project.
Okay, here's another question. In one of the slides, it showed a chart that depicted the difference in how dogs barely die of cardiovascular disease and that humans are much more likely. Could this be because dogs have a much more controlled diet while humans sometimes tend to eat unhealthy foods like fast foods, which leads to problems?
What a great question. So I would say yes and no. Um, there's a really cool study that came out pretty recently, a year or two ago, where anthropologists went down to South America and studied an indigenous population that until recently has lived in the Amazon like it always did. It's a population called the Tsimane, Tsimane, and they have almost no heart disease. It's stunning how little heart disease they have. And they're very active and they have a healthy diet with probably very little saturated fats. So I think that's a big part of it. But the other piece of it is that it might be part of the mechanism is that the veterinarians tell me that dogs metabolize cholesterol in a very different way, such that they don't get the kinds of arterial plaques clogging up the arteries that we do. So probably, your question is a really good one. It's sort of an either-or question, and the answer is probably that it's both diet and something inherently biological.
Is there a notable difference in the statistics of age-related conditions between purebred and mixed breed dogs?
We now have the data to ask that question. Until now, the only thing that people have really looked at is lifespan. And we know that mixed breed dogs on average live about a year longer than purebred dogs. But it's going to be really fun to look at this. And I'll just suggest one really interesting population that I'm excited to look at is what they call the designer breeds, and the most common of which is the Labradoodle, where people take two purebreds and cross them together, and there are thousands and thousands of Labradoodles in this country. I don't know how many are signed up in our study, but it will begin to help us determine how much of disease profiles are due to just being inbred and how much are due to the particular genes that are found in, let's say, a Labrador Retriever versus a Poodle. So it's a question that we can already start asking with our survey data, and I'm excited to ask them. We don't yet know the question. We don't yet know the answer.
Okay. As a non-dog owner, how can I be involved to help in this study?
Yeah, great question. Thank you for asking that. So in lots of different ways. Once the data are fully released, we will welcome people to look at the data if they have the wherewithal to do statistical analysis. We also welcome people to help us get the word out and tell your friends to sign up. We are still recruiting dogs. We actually are just starting a second big launch. There are holes in our data set. So for example, because of COVID, between the time of our big nomination launch in 2019 and when we sent the surveys out, all of those dogs that were in their first year of life became 1-year-olds and older. So we don't have enough puppies. If you know people with puppies, tell them to sign up. We don't have enough rural dogs, for example. So we're excited about having people help us recruit, help us get the word out in that way. We don't have labs full of dogs because all this research is observational, watching people's dogs in their homes. But there are labs doing the research. And so if, for example, you're an undergraduate thinking about graduate school, there will be lots of opportunities for study for students to join the project.
Or do you have internships with the projects? Are you thinking about doing things like that?
So I've mentioned funding a couple of times. One of the things on my list that I would love to find philanthropic support for is to create internships at all levels, high school, undergraduate, and graduate. the budget we have is large, but because our numbers are even larger than our budget, we don't have a lot of funding for internships. So I'm really excited to try and create those. That we're really excited about the educational broader impacts of the study, and that would be one of them.
Great. Why can a dog be part of the pack if its heritage is completely unknown?
So every dog, so first of all, the one of the top predictors of what's going to happen with a dog is its size. And so even if the genetics of a dog is unknown, we know its size. The second thing, this is such a great question. So think about it this way. What we really want, so we're hoping to learn things that will teach us about human aging, but we also want to learn things that will teach us about dog aging that would be good for dogs. And so many dogs have an unknown ancestry, and we want to be able to understand aging in dogs of unknown ancestry. And the best way to do that is to study dogs that don't have a known ancestry. That said, and sorry about the barking dog across the street, you might be able to hear it. So that said, many of the dogs, thousands of the dogs that have unknown ancestry will be fully genome sequenced. And from those sequences, we'll actually be able to know exactly what proportion of the dog comes from which breed.
Do you have researchers involved in the project doing qualitative research beyond ethical questions?
Yeah, so I hope we do, and I'm just going to tell you a story to illustrate what's possible. So, in terms of the ethical questions, we have a team ethicist who's a pediatric MD PhD ethicist who is the sort of house ethicist at Seattle Children's Hospital. And then we also have an independent ethicist who's a veterinarian and the head of our Animal Welfare Advisory Board. They're actually interested in studying questions, qualitative questions like, why are people electing to put their dogs in a clinical trial? They're dogs who are healthy. Why would you choose to give a healthy dog a drug? Our goal is to keep it healthier longer, but we want to know what people are thinking. But I'll tell you another story. So I got an email a while ago from a woman who's a professor in sociology, and she's really interested in studying the sociology of older couples without children. And she really wants to know, what is the role that pet dogs play in the lives of older couples without children, and how that affects their own lives, how that affects their relationship with other couples that might also not have children with dogs, and how does the presence of a dog in that home, in that social circle, affect their lives compared to couples without children who don't have pet dogs. That's one of just a million ideas. So yes, lots of interest in qualitative studies. And so I kind of glossed over the last slide. So we actually are writing a paper right now that will be, we hope will be in a high profile journal to sort of announce us to the world. And one of the things that we'll stress in the paper is that we welcome ancillary proposals. So our hope is, for example, that my sociologist colleague here at the University of Washington will contact me and say, I want to submit a proposal to the National Science Foundation to gain ancillary funding to study 100 couples who are in the dog aging project. And so those kind of ancillary studies we welcome. Part of our impact is by making research possible by people outside of our team for these ancillary studies.
Are you also tracking the lifespans and morbidity of the owners?
So I have to say this this audience is asking just all the best questions. So we're really excited to do this. So when you start asking people questions, and I'll explain this a little bit for the benefit of the undergraduates here. Anytime you do research with animals, if the animal is a vertebrate, so it's not true with fruit flies or worms or fungi like yeast, but if you're studying vertebrate animals, before you can even begin to collect data, you need to get approval from a panel of expert researchers at your institution, which is the Institutional Animal Care and Use Committee, or IACUC. And so we have IACUC approval both from Texas A&M and from the University of Washington for the dog aging project. As soon as you get into people, the bar goes up, and you need to get approval from a different panel of experts that's called the Institutional Review Board, or IRB. If I want to ask you, so there are questions that we can ask without IRB approval. So for example, we ask, is the dog in your home exposed to secondhand cigarette smoke? We're allowed to ask that. We're not allowed to ask, do you smoke? So we can't ask anything about the people. Ultimately, one of the things that we're really interested in is the notion that dogs might be what we call sentinels. So if you think about the canary in the coal mine, so canaries, it's a real thing, the canary in the coal mine. Canaries are very sensitive to oxygen levels. When carbon monoxide levels go up in a mine, the canary would pass out before the miners would notice. And so the canary passes out, they hustle out of the mine. So that's a sentinel. So we think that dogs might be really valuable sentinels for environmental risk factors, because dogs spend more time in our environment than we do. Dogs stay home when we go to work. And dogs are much shorter lived, sadly, than we are. And so everything is sped up. So there might be risk factors that are impacting our aging trajectory in a negative way that we won't notice until we see it in dogs. To study that, we're going to need to track the owners and the dogs. So this is a big lift for us. We're excited to do it. Once the dog part of the project is fully underway, then we'll start moving into the owners as well. So long-winded answer is yes, but not yet.
I think that's all the questions I see. Does anybody else have any questions to throw out? Yes, no? Well, if there's not any more questions, thank you very much. That was really super interesting. I appreciate it.
Really a pleasure. And let me say, if anyone here does have questions that come to mind afterwards, please feel free to reach out to me. My email is Promislow without the W, so PROMISLO @uw.edu. And if you just tell me that you were at the seminar and you had a question, don't hesitate, and I'll be happy to do my best to answer it.
Great, that's awesome. Okay, last chance for a question. All right, I'm not seeing one coming in.
And to the students here, good luck with class registration.
Okay. All right. Well, thanks so much.
Thank you again, Dr. Manis, for the invitation. I really appreciate it.
Yeah. Thank you. All right. Take care.
Bye-bye.